speaker. Jim and I taught genetics in this room. I stood up and talked in here for a long time. I met Jim in 1984 at the Colloquium recombination meeting, where he asked me something about RecF, I remember, over lunch. And I actually considered working with Jim as a postdoc. I went to Bob Warner's lab. I was wanting to work on yeast recombination. But I didn't know where Waltham, Massachusetts was. So I was like, where the hell's that? Jim is responsible for recruiting me here to Brandeis. He's been renowned for the study of homologous recombination by the use of physical techniques to detect transactions. And he was doing that in 1984 when I met him. And he stuck with it. It's sort of Hershey heaven for Jim. Physical monitoring for combination, and it turned out to be important for meiosis, double-strand breaks underlying meiotic recombination, and predisposition to cancer is associated with failure to repair DNA. So Jim is very well known in the recombination DNA repair cancer community for his contributions to our understanding of eukaryotic double-strand break repair. He won the Thomas Hunt Morgan Award from the Genetic Society of America in, I think, 2010. So it was a Lifetime Achievement Award, and he's had another half of a lifetime after that to continue his contributions. He's a member of the National Academy, all the academies, the American Academy of Arts and Sciences. And we've been lucky to have him here for had a sort of big birthday recently, but this is sort of a celebration of his career here at Brandeis. So thank you.
It has been a 50-year pleasure to be here. So I have roughly 50 minutes to talk about a 50-year career, which is 1 minute per year. And you'll forgive me if I sit, because one of the things that neuroscience has not achieved yet is the relief of chronic pain. Anyway, about 100 years ago, I just wrote an article in Genetics about a paper that was published 101 years ago that saw how DNA had to be structured. But also about 100 years ago, a poet named Wallace Stevens wrote a wonderful set of poems called 13 Ways of Looking at a Blackbird. And so my alternative title is this. I'm going to tell not 13 stories, but a number of interlocked stories about DNA repair. And in each case, there are still elements of the black box that we really don't understand. So first, just a very brief word about my evolution. I went to college, I went to graduate school, I came here. Okay, I've been here. And since I've been here, we've worked on really we have branched out and done things in quite a large number of different areas. Obviously, I'm not going to talk about all of them or even a few of them. I'm going to talk about homologous recombination. But the new thing is to invade Bruce Good's world, is the role of actin and myosin in homologous recombination, which is a new and big surprise. So, okay, so I came to Brandeis from my postdoc, never having had a genetics course, and I got here and the chair said, "You'll teach genetics." I said, "I don't know any genetics." He said, "Here's the book." And I learned it by teaching it. But my salvation and my inspiration came a couple of years later when Jeff Hall joined the department. Jeff, of course, getting the Nobel Prize with Michael. But Jeff was a master of classic genetics. He knew absolutely everything about all the old papers, and he loved to teach them. And what he taught were all the classic people who had done fly genetics, but especially about Barbara McClintock, who had a very profound influence on my work, and with whom I formed a friendship that lasted for about 15 years. And Jeff, and so this is the thing, I was Jeff's best student. Okay. Students in the class, yeah, they got some of it. I really paid attention. And so a few years later, Jeff stopped teaching this course, and by then I had absorbed enough so that I could really teach it with some degree of authority. And we met, oops, that's not.
That's Jeff, just put that up there.
The host would like you to unmute. Am I not unmuted? Is that for somebody else? I don't know how to get rid of that.
The computer is next to you.
But I can't do anything until it goes away. So,
Click on mute.
Click up in the. No, I think that isn't there another computer?
You can just click on mute, probably.
There's a mouse right next to it.
Or hit enter or something.
I have to do something else.
I need something that's you can just say, yeah.
Okay, so Jeff, me, best student, and we met Barbara. I met Barbara McClintock. She came here in '77, and then it turns out that I really paid attention to Jeff's stuff. And one of the things that Jeff taught was this really arcane set of experiments, first done actually in flies by somebody named Lillian Morgan, who was Thomas Hunt Morgan's wife, but then done by McClintock. And the idea was that she had isolated a circular version of a chromosome. And when that circular version of the chromosome recombined in meiosis with a linear version of the chromosome, it made a dicentric chromosome. And this is subject to being yanked on by mitotic spindle, and it breaks. At least in maize it breaks. In flies it turned out it didn't. And then this broken piece could undergo a whole series of what were called breakage fusion bridge cycles, but eventually end up in what McClintock called healed chromosomes that had new ends, new telomeres, and were stable. And so in 45 years after she published this, we accidentally made a ring chromosome in Saccharomyces while we were studying mating type. And probably the only yeast geneticist in the world who would have said, oh, we can do the McClintock experiment, because Jeff had taught me. And so we did the McClintock experiment. And what we found was that the forces in meiosis were surprisingly different from the forces in mitosis, and there was no breakage of this dicentric chromosome as it entered into a spore. But once it was in a spore, and when underwent replication and then mitotic segregation, there were lots of breakage events. And from a single one of these spores, we could get as many as 9 different outcomes. And among those outcomes were new chromosomes, rearranged chromosomes that had new ends. And that's really the beginning of how I got interested in break repair. Because at the time, mating type switching was not known to be a break repair type mechanism. And so for the next whatever years, we've been studying these two things, how cells fix their broken chromosomes and how they know that they have damage. And here's the obligate Haber lab slide that everybody shows. You have 30 trillion cells in your body, and virtually every cell in your body has the same karyotype. Your genome is incredibly stable, except when it's not. And when it's not, in tumor cells, you can see 100 chromosome rearrangements. And so these cells have lost their ability to do accurate repair, to restore the genome to its original state. They're not without the ability to repair because they do end joining incredibly efficiently, which is why you end up with all these rearrangements. But they've either lost their ability to do homologous repair, which are genes like the breast cancer susceptibility genes BRCA1 and BRCA2 and lots of other cancer susceptibility genes turn out to be defects in homologous recombination, or they have defects in the checkpoint, which stops cells from dividing and eliminates them so that they don't propagate. And so for both of those reasons, cancer cells are the result, and we're interested in both of these processes. But I'm going to talk really just about the first one. Okay, and the second thing is that the source of these breaks, for the most part, is just our normal process of DNA replication. That when cells replicate, even though they are incredibly accurate at one level, they have to replicate this much DNA, okay, and they make a dozen mistakes of a particular sort, so that if you eliminate the recombination protein RAD51, you can see these chromatid breaks where there's one intact sister chromatid and a broken one. And the broken one is going to use the RAD51 protein to find its partner and copy what's missing to restore this process. And that's basically the processes that I'm going to talk about. Okay, but even in your cells that don't divide, like in your brain, it turns out thinking caused double-strand breaks. Okay, and this is this beautiful paper from Mike Greenberg's lab, Sue Parady's previous advisor, that really shows the appearance of activity-dependent double-strand breaks in the brain, which is really an astonishing result. And it means that these processes are, and they showed that mice that don't have these abilities to repair die early. Anyway, so there are all these different ways of sort of repairing a break. You can put a new telomere on the end, as we have shown. You can join these together by what are called non-homologous end joining mechanisms, and there are at least two very distinct mechanisms. And there are these homologous recombination mechanisms, which look for homology in order to have to use the ends of the break to find a partner and patch up the break with either a little patch or a long run of new DNA. And these are two very different processes, and I'm going to talk about this one, the gene conversion process, and I'll also talk a little bit about this break-induced replication process. And they both start by the ends of the DNA in cooperation with RAD51, finding a partner and forming Watson-Crick base pairs to establish the fact that this is a place to do the copying, to do the repair. Okay, so I got very interested in the mating type switching system in Saccharomyces. We were interested in it before we knew that it was initiated by a double-strand break, but we especially have been interested in it since. And the thing about mating type switching was that very efficiently a cell of one mating type could switch to the other mating type or back. And it became evident after a while that the sequences that specify one mating type are totally different from the sequences that specify the other mating type. And in order, therefore, to switch from one mating type to the other, there had to be a replacement of one set of sequences from the other. And the replacements depend on two donor loci, called HML, HM left and HM right, and it stands for homothalasm, which turns out to be the process by which a single cell can turn itself into a diploid. And this process is initiated by a double-strand break, by a site-specific endonuclease that cleaves the DNA here, and then there's a recombination process that removes the red sequences and puts another copy of the blue sequences. And even though there's an equivalent site here that HO ought to cut, or an equivalent site here that it can cut, but not here in the donor, it's because these donors are heterochromatic, silenced by a highly positioned nucleosomes. And as a result, they also can't be cleaved by the HO nuclease. And one of the mysteries not solved is how do the ends, the broken ends of DNA, invade exactly the same place that the HO endonuclease can't cut? And how does it get in there to do the recombination in the first place? And that's really not understood. But we know it happens. Okay. That's what that says. Okay, so the big breakthrough for us, as Sue said, is that we started to be able to physically monitor these kinds of events. And the idea was to use southern blots and to use a galactose-inducible version of this nuclease so we could turn it on. And since the restriction sites are different in this fragment than in this fragment, we could see that mating type switched from one mating type to the other, and we could see that it got cut. And it was cut incredibly efficiently. But we also learned that it was slow. And there were lots of slow steps in this process that we could follow. And so we followed them by various means, by PCR and southern blots, by chromatin immunoprecipitation, more recently, as I'll show you, by live cell monitoring. And we could sort of follow all the steps in this process. And of course, we in this case does not mean just my lab. There were other people, lots of other people involved in this, but we had a big role in it. And so the first thing that happens is there's a break, and then there's a whole series of exonucleases that chew away the end to leave a long 3'-ended single-stranded piece of DNA. That piece of DNA gets bound by the Rad51 protein. Every 3 nucleotides, a filament is formed. And this filament has this capacity to sort of bang around the genome, find homology, pry open the DNA, and form those Watson-Crick base pairs. And that intermediate structure, which is called a D loop, then serves as a recruitment site for new DNA polymerase. And DNA polymerase starts to copy this. But it's not like normal DNA replication, because you probably remember from the famous Messelson and Stahl experiment about heavy and light DNA that all the DNA in replication is semi-conservative. It has one old strand and one new strand. But here, this strand is displaced as if it was RNA. Okay? And so the donor ends up being unchanged, and this newly synthesized strand gets displaced, and then the second end gets captured. And I'll talk about the capture a little bit. And then a new round of DNA synthesis happens, and all the newly synthesized DNA ends up in one place. And that turns out to be not like normal replication, and it turns out that the fact that this is different leads to a very, very surprisingly high rate of mutagenesis in these sequences, that the normal quality control mechanisms that are part of DNA replication don't work well in this context. Okay, so I'm going to talk about 3 aspects of this, some old stuff and some brand new stuff mixed together. And I'm going to use silent movie stills like this to tell you that I've changed subjects. So if you get confused, wait 5 minutes, it'll be something else. And there'll be a quiz at the end as to who all these people are. Okay, so Buster P. Anyway, the first one I want to talk about is actually the second one on that list, the search for homology. And so in the teens, it became possible by using formaldehyde cross-linking and PCR methods, or DNA sequencing methods, to figure out how frequently any 2 sites in the genome banged into each other, all of which are very rare events, but you could find them, and you could computationally then make a picture of how the chromosomes in yeast were organized. And all the centromeres we already knew were all clustered at one end, but there was this picture. But the question really is this a static picture, or does it have some real meaning in terms of mechanism in the following sense? If you have a break here, does it matter whether the donor is here or there, excuse me? Or is this really just a static picture of something that is so mobile that it really doesn't matter, all the sites are equivalent? And the answer is that all the sites are not equivalent. And so Cheng-Cheng Li, when he was a student in my lab, put a copy of a gene, LU2, out near the end of one chromosome, and then he put the donor sequence, just a 2 kb piece of DNA, elsewhere in the genome in 20 different locations, and said, how efficient is this repair event? And the answer is that the efficiency is very much a function of the contact probability, the likelihood that these 2 sequences have banged into each other. So, and we could change that in a variety of ways. More homology was better, and lots of other things were better. But there was an intrinsic position effect in terms of how recombination was going to take place. And I'll come back to this in a minute. Okay, so the second aspect of this is what's happening to the DNA itself. And a number of labs took the system that we were using and, as we had done, removed these 2 donor sequences. So now there's a break that can't be easily repaired because the donors are gone. You can still do end joining, but it doesn't do it very often. And so mostly now we have a broken chromosome where the ends are being resected and it goes on resecting and resecting for a very long time. Okay, and so Susan Gasser's lab took a version of this and put a fluorescent marker somewhere on a chromosome and showed that there was an increase in chromosome mobility, which is measured by something called mean squared displacement. And the increase in, and these are actually data from Felix show in my lab, but the idea is that normally the chromosome is moving at some rate, and now it's exploring more space. And that would suggest that this is an important aspect of finding homology in the genome. And so this is Rod Rothstein's lab picture, that the same sites after the cells were fairly heavily irradiated showed much more mobility. Okay, and so what we sort of look at is where they plateau this radius of confinement, which is sort of a limit to how far these sequences can sort of move around in the nucleus. Okay, so lots of labs, mostly Susan Gasser's and Rod Rothstein's, showed that to get this increase, you needed an activation of the DNA damage response, which I'm not talking about. You needed chromatin remodeling around the break site, but it also caused motion in sites that were not on the same chromosome. So there was a global increase in chromosome movement, which is actually what is being measured here. But what I'm going to add to this is that actin and myosin turn out to be key players in this, which we have not appreciated. So the first indications of this actually came from studies in mammalian cells, where somebody, Jean Gautier's lab, used a drug called CK666 to inhibit Bruce's favorite protein, ARP23. And what does ARP23 do? It catalyzes branching of the actin filaments. And in yeast, it is predominantly used to promote endocytosis. And a lot is known about that from David Drubin's lab and then Bruce's lab. Jeff Gellis's lab has participated as well. And so we know that there's something about endocytosis here, but what does this have to do with the nucleus? Okay, and so that's the question. And what Felix then went on to do, and I should say he motivated this entirely by himself. Okay, I wasn't sure this was going to be a productive direction. And all credit to Felix for sort of learning all its techniques and going ahead with this. And so what Felix found was that when you added CK666 20 minutes before you did the monitoring of the motion is inhibited. So that was interesting. And then the question is, what other components in the system are needed? And the answer is all of them. Well, not all of them, because things that are specific for endocytosis and not part of this mechanism are not needed. But the mechanism of these nucleation promoting factors turn out to all be important. Okay, and so come to Felix, because there's a lot more than I'm going to talk about. Okay, so one of the ways that we've taken advantage of new technology is by what are called auxin-inducible degrons. And somebody figured out, Kanemaki's lab figured out that if you put a little piece of a plant protein that binds auxin, which is also called indole acetic acid, onto the end of any yeast protein, and you brought in a specificity factor that also comes from Arabidopsis, that the combination of these two things, when you add auxin, put this protein in the vicinity of a ubiquityl ligase, that got ubiquitylated, and then it went to the proteasome and it was degraded. And so in an hour, you can degrade the protein. And what's wonderful about this is that the cells are basically normal until just before you do the experiment. So there are no suppressors arising from having gotten, disabling endocytosis or other things. And so this turns out to be a really terrific way of inactivating genes in yeast and in other organisms too. Okay, and so if you inactivate LAS17, which is also called WASP, and you see the same problem. If you inactivate myosin 1, either of the two myosins, you see the same problem. So all of these factors cause some reduction in chromosome mobility. And but if you inhibit endocytosis in other means by a gene called SLA2, it doesn't have any effect. This seems to be something happening in the nucleus. And I'll tell you now, before you ask, no, we have not physically shown that actin is in the nucleus doing this. Okay, it's very hard, turns out technically hard to do. We're not the only people who haven't done it. So I'll come back to the question that I started with, which was, how do these cells find their distant donors? And we made the hypothesis, I think a reasonable one, that if distant donors are hard to find, that slowing down the motion of the broken ends ought to have a consequence, which would be that the distant donors would really have a hard time being found. And the answer is not. Okay, so comparing these different locations, moving this donor to different locations, either in the wild type or in the cells lacking MYR5, which has this much lower radius of confinement, there's no difference. And so this, in my view, is still a phenomenon in search of a function, because we really don't see it in the ways that we have looked for. Okay, slightly change the subject. The resection part of this. It'll turn out it's also about actin. Okay. So a long time ago, Charles White in my lab figured out that he was the first person to show that there were intermediates of recombination that he could detect. And he could detect this one because it's the nucleases chew down the DNA and fail to cut. When they chew past this, single-strand DNA can't be cut by this restriction enzyme. So that means instead of getting this fragment of DNA, you get this fragment of DNA. And Charles showed that he could see these intermediates. More recently, there are other better ways of doing this measurement, as I'll show you. And so it turns out there are 3 different nucleases, or sets of nucleases, that do this removal. The early part is done by a complex involving these proteins, one of which is called emery-11. And then there are 2 what are called long-range exonucleases that chew away the DNA. One's actually an exonuclease. It chews off one nucleotide at a time. The other is actually a helicase that unwinds the DNA in an endonuclease, so it's lopping off little chunks. But they both have the same consequence, which is that they can go down the DNA. They go down the DNA quite slowly. They go at 1 nucleotide a second, 4 kb an hour, and it turns out they'll do it for 24 hours. Okay, I mean, this is a mindless process once it gets started. And so the second thing that we found is that there's another protein, a chromatin remodeler, called SWI-FUN30, and it turns out to be needed to do both of these processes. And we think it must be unwinding the DNA around nucleosomes so that the process can get down the DNA, which of course is covered in nucleosomes. And so we can slow down the resection either by disabling these nucleases or by getting rid of FUN30. And what Felix showed was that when you slow down resection, you slow down the motion of the chromosome, which we do not understand. Okay? And that's true also even when the nucleases are functional, but you've just gotten rid of this chromatin remodeler. And so among the many questions then is, what's the correlation between this kind of resection activity and the increased mobility of the chromosome ends? And we don't know. Okay. The second thing that Felix found was that if you applied this drug CK666, or if you inactivated these other proteins before you made the double-strand break, we didn't see foci. We didn't see the appearance of these fluorescent proteins bound to the end of the DNA, and that was surprising. But the HO was cutting. That was critical. So the cut's made, but there are no foci. And the reason there are no foci is there's almost no resection. Okay, and so this actin-myosin machine is controlling resection even from the very beginning. Okay, and so these are preliminary data from Marissa in my lab and Kevin, who's a rotator, just showing that if you apply if you inactivate LAS17 either before or after the HO break, that a site that's about somewhere about 5 kb away from the end of this break is basically not resected. This is actually a PCR assay that looks for the preservation of this sequence rather than for what Charles did, which was a different measurement. But it gives you the same result. Okay, so how does this work? How does ARP do this? And conversely, how does resection affect mobility? And those are questions that we haven't got answers to yet. So that's why I'm still here. But one thing that other people have done recently is to suggest that this WASP protein has a direct interaction with the single-strand DNA binding protein, RPA, which is needed for these other proteins to bind. And so we're very interested in how RPA fits into this story, but we don't know yet. Okay. So now I'm going to turn subject to back to the sort of mechanism of this repair, and I just remind you that this process is different from normal replication because all the newly synthesized DNA ends up in one place. And now I have to introduce you this other version of this process called break-induced replication. It happens when there is only one end, such as the telomeres, which only, of course, are at the end of the chromosome. And it turns out that this mechanism is needed to maintain telomeres in the absence of telomerase, which is a cancer condition called ALT. And what we first showed, and was taken up by other people, Roger Greenberg and others, to show that these same things that I'm talking about are applicable to mammalian cell chromosome maintenance. Okay. And the other time this happens is if this process fails to capture the second end. And since these processes start out the same, it turns out that they are interconvertible for some period of time. And I'll show you that. Okay, so again, it's unlike normal DNA replication, and it's very error-prone. Okay, so I'm not going to show you any of the data for these results, but we've looked for how, using physical means, how different mutations affect the ability to do these repair events. The little patching up event doesn't need much of the DNA polymerase apparatus. But the break-induced replication process needs everything, essentially, that's in the normal DNA replication fork, and more. And the more turn out to be two proteins, Pol32 and PIF1. We showed that this one we first showed this one, and then it was shown in mammalian cells that it's needed for ALT, the same homolog. And Greg Ira and Anna Malkova, who used to be postdocs with me, showed that the PIF1 helicase was also a critical part of this process. And this process is different from normal DNA replication also because one strand gets manufactured and only later does a second strand get added. And it turns out they're both added by the same polymerase, DNA polymerase delta. And that also is not normal in terms of normal DNA replication. Okay, so in normal DNA replication, Pol delta just does little snippets. It does what are called Okazaki fragments, a few hundred base pairs in length, and does them over and over again as the replication fork proceeds. But the leading strand is done by Pol epsilon, which can go a long, long way without interruption. And we took advantage, and the question is, well then, why do you need PIF1 and Pol32 to do break-induced replication, but not for normal replication? Okay, and so we took advantage of a mutation called Pol216 that somebody had made a long time ago, which keeps the structure of Pol epsilon. We don't know why it has to be there, but it does. But it's catalytically dead, so that it's Pol delta that's going to have to do the actual polymerizing on this strand. And it turns out that if you look at this weird condition, you need PIF1 and Pol32. So in order to make Pol delta processive, to go down the DNA a long way, and not to do just Okazaki fragments, we think that's why you need these two proteins. And that's as close as we've come to an explanation. Okay, so now I'm going to talk about the third of these things, second-end capture. And in second-end capture, we know that the break starts here and starts copying in this direction, so the second end it gets captured over here. And Anuja Mehta, who was a grad student in the lab, said, what happens if I just shorten the homology progressively on this side? What does it do? And it turns out it has a huge effect. And when you get down to 50 base pairs, there's almost no recombination at all. At least only a few cells are alive. But it turns out there's lots of recombination. It's just that we didn't know it until we physically looked. And the lots of recombination is break-induced replication, where this end is invading and copying all the way to the end of this chromosome, but that's lethal. Okay, and so the cells die, but they're making a product. And we proved that it's the BIR product because if you knock out Pol 32, it's gone, but you don't do anything to the other event. Okay, so I just say we also have learned how to push this around a little bit. There are two helicases, proteins that unwind DNA, and they have opposite effects. So if we knock out one of them, we can push this balance towards nothing but the gene conversion event. But if we knock out the other one, we can push it almost completely in the other direction. So we begin to see how these processes are interconvertible and how these helicases play a role in this process. And that's can be sort of re-summarized to say that this is a very delicate process. It's very different from normal DNA replication, where everything's held together in a very stable fork. But here, there's this dissociation event, which is going to eventually end up in second-end capture. But if the helicase unwinding this goes faster than the polymerase, it may interrupt itself before it's ready to really accomplish the end of the recombination. And so when it does that, maybe it can reinvade, but it also turns out it can go elsewhere. And so I'm going to tell you about going elsewhere, that we see lots of illegitimate events that go to wrong places because this dissociation is happening too frequently. Okay, and the agent for doing this is this DNA polymerase delta, which is interesting. It's an amazing set of proteins because these proteins can completely change their conformation to do what is called proofreading, to remove bases that are in the wrong base that was put in. It can remove them by an exonuclease activity. But there's a mutant called Pol 3-1 that I'm going to talk about, which doesn't do this transition. And it turns out, I'll just say it now, we believe it to be more processive. It doesn't fall off the DNA as often. It turns out, ironically, it makes lots of base pair errors because it can't fix them, but it doesn't do any of these weird events that I'm going to show you. Okay, and I wrote a haiku in case you want to remember. That's a summary of the rest of it. Okay, so even gene conversion turns out to be mutagenic under these conditions, and in fact there's a 1,000-fold increase in the rate of mutation of these same sequences that are copied during repair as compared to when they're just replicated. Okay, so to do this, Wade Hicks, who was a very creative grad student in the lab, tricked up the mating system by replacing the normal mating type sequences that are here in the donor locus with a copy of URA3 that we got from somebody else's lab, and it's not from Cerevisiae, it's from another yeast called Colloquium, and that will turn out to be very surprising and interesting. Okay, so these sequences are silent, as I told you, they're held in the silent state, so even though this is a functional copy of URA3, the cells are URA-. When you turn on HO, you replace these sequences with a copy of these sequences, so now they're URA+. That's great. And you can select for mutants, because we have a drug called 5-fluoroerotic acid that will select for URA- mutants. And to show that these are new mutants, we unsilenced the silent copy, and these URA- cells become URA+, because this copy is still wild type. And that means that the event that happened during the switch. And those are the things that we now look at. Okay, and so we've done lots of studies on this, and I'll just, template switching, which is the motif I'm going to talk about, is that the polymerase is falling off, or the newly synthesized strand is falling off and getting back on in some mutagenic way. And so it turns out lots of these events have what I'll call a signature. They make small frame shifts. They make something called quasi-palindromes that Sue Lovett had a great deal of a big role in characterizing, and something else that I'll show you in a minute. So we sequenced lots of these. These are early sequencing results. Everything above the line is a base pair substitution. They're fine. We're not interested in them. Okay. The things below the line are changes in length. And the most obvious change in length are these -1 events. And here's 3 Gs become 3 Gs become 2 Gs. Here are 4 Cs become 3 Cs. They're all minus. And that's interesting because in replication, a sequence like this is just as likely to add a base as it is to subtract a base. And yet in the repair, they're almost all minus. And then again, we don't know why, but we think that it means that the machinery itself is fundamentally different. Okay, that there's something about the way in which the polymerases are moving down the DNA, which, if you want to, for want of a better word, have forward momentum. They can't go backwards. Okay. Okay, so those were interesting. And then there were weird events. Weird events that changed both the length and some base pairs. And how Wade saw these, I have never figured out. But these are called quasi-palindromes that Sue knows about. And a quasi-palindrome is a sequence that can self-anneal. And it isn't quite a perfect palindrome. There are base pair differences in it. But in order for this to happen, that means that this newly copied strand has to fall off from its template and bind to itself. Okay? And then it elongates, and then it has to unwind all that and go back where it was and finish the process. Because only if it gets all the way to the end do we recover the events. Okay? So it turns out that these are quasi-palindrome events. And so recently, Sue helped design, and in collaboration with her lab, with Mason, we have been studying these very specific events by making a specific quasi-palindrome in the sequence. It has a 4 base pair insertion. That means that the open reading frame is frame shifted and it's Ura minus. But if it goes through this process that I just showed you, you can actually end up with Ura plus cells. And that turns out to be that all of the, 20 out of 20 of the events that we recovered were changes in the quasi-palindrome. So we have a really nice system to look specifically at these kinds of events. And we have similar substrates to look at frame shifts and other things that we're interested in, that we sort of broke it down. And so I'll just say one thing about this one, which is, as I told you, Paul 3-1, the proofreading defective guy, doesn't do these events very well, and so you can see that here. And there's obviously roles for mismatch correction, which are these 2 proteins that I'm not going to talk about. Okay. So this has been a great collaboration with Sue's lab. And then finally, the last set of these things I want to talk about are completely weird events because they have DNA from somewhere else. Okay, and it turns out it isn't just randomly somewhere else, and it's a demonstration of how you can be lucky and lazy, or lazy and lucky. We knew this sequence came from Clover-Myces lactis, and we knew that Cerevisiae had such a sequence for the same gene, UR3, but it's only 72% identical. It's incredibly diverged. And it's not expressed because there's a transposon sitting here. Okay. And we said, what could possibly go wrong? Okay. Well, it turns out that these crazy events, those sequences that I just showed you, come by the fact that as this is being copied, as this is being copied, it jumps to this other chromosome, picks up sequences, and goes back. And it has to make 2 jumps in order to do this, because only if we get to the back to this sequence can we put all of that stuff in. So these events not only dissociate, but they go to a completely different chromosome, find a sequence that is only 72% identical, copy part of it, and go back. And we would never have seen these events if we had been more careful, I think, and said, well, we should get rid of this. We should get rid of this before we do the experiment. Then we never would have seen it. So we were lucky. And so Olga Cepelino, was a postdoc in the lab, turned the system around and said, let's select for those events. She put a 32 base pair deletion in this sequence so that if it switches, the cells are still Ura minus. But if it does this jump, we can get Ura pluses, which would be chimeric genes, partly Clover-Myces, partly Saccharomyces, and they're functional. And so that's what she found. They happen fairly rarely, but if we make these sequences identical to these sequences, there's an almost 10,000-fold increase in the frequency with which these events occur. They're now occurring in 1 in 1,000 cells, or a few in 1,000 cells. And that's amazing, because that means that this repair polymerase is just incredibly unstable. It's falling off, the sequences are going somewhere else, picking up more sequences and going back, which is just never happens in a normal replication field. This is a completely different apparatus. And so we also have found a couple of genes that are not needed to do this simple process, but are needed for this, and we don't yet know much about what either of them is doing, but we're still working on it. Okay. How much gets copied from the second location is variable, and if you look where they go in, they always go in at a place where there is microhomology, but they go into many different microhomologies. And if you choose one of those microhomologies as where it jumped the first place, it goes back in many different locations, so that there is no fixed beginning or no fixed ending to this process. And we can sort of look at all of these things, and the longest microhomologies are used the most, and Carmen Macias, as an undergrad, is asking, well, what happens if we just get rid of one base and make this into 2 shorter microhomologies, because when there are 2 shorter microhomologies, they're not used so much. So we're in the process of trying to figure that out. But we don't know the rules, because there are some very long microhomologies not used very much, and there are other rather short ones that are used a lot, so we don't know the rules. But they're important rules, because it's been very clear now that in mammalian cells there's a similar process going on called microhomology-mediated something or recombination of some sort. And so this is a model for really trying to understand how these events are happening in mammalian cells. Okay. So, as I said, almost all the -1s and these other events go away when you get rid of the polymerase with the proofreading activity. And there's an old piece of biochemistry that sort of argues why that might be. And this was done by Mike Resnick and Dmitry Gordini and Peter Bergers. This is in vitro. This is an end where a polymerase can bind, and it will go down the DNA, but the wild-type enzyme gets to here and stops. The mutant enzyme goes all the way through. Okay. So what we think that means is that the proofreading defective enzyme stays on the DNA, doesn't fall off as often. Therefore, the frequency of all these crazy events, which require some kind of dissociation, are reduced, even though it's making many more base pair errors. But we think that's a good explanation for why the proofreading polymerase is in fact, an anti-mutator in this particular context. Okay, so we've done lots more sequencing, collected lots and lots of mutants, and I'm just going to finish up by talking about one other set of these mutants, and that is we looked for intramolecular, intragenic deletions or tandem duplications. So tandem duplications would just take the same sequence and duplicate it, and you can see how that would happen. You're copying down the DNA, it dissociates, but it now goes to this microhomology and starts up again, so you make a deletion which is bounded by the microhomology. And conversely, a tandem duplication would go all the way to here, fall off, go back to here, and start up again, and you get it. Now you get a tandem duplication. So we've looked for those events, and it turns out that there are many more deletions than tandem duplications. And there's one other thing that's very strange here. There are lots of deletions, but there are no small deletions. So less than 25 base pairs, we don't see any deletions. And so that's very curious, and the way we're interpreting this is sort of my, geneticist picture of what's going on at the replication fork, is that as the DNA is being copied, and there's this dissociation event, there's a helicase coming behind which is closing up the DNA as fast as it's being copied. And so these sequences are no longer easily available. They're now in double-stranded DNA. But ahead of the fork, there's another helicase opening up the DNA. And these sequences here are available as single-stranded DNA, where we can make lots of deletions, but therefore will be very difficult to make tandem duplications. And we've now built a deletion strain, we're building a tandem duplication strain of the same sort I talked about, so we can look specifically at those kinds of events, but we haven't done them yet. And then the other question is what's sitting here? Because something is precluding the first 25 base pairs from working in this context, and we don't know, again, what that is. Okay. So there are lots of questions. I haven't even raised the ones about how a nucleosome is put back in this. How does this DNA get restored back to being normal chromatin? How is that related to the processes in normal replication? That's something we really ought to find out about. And again, how is break-induced replication different from normal replication events? Okay, so this is just one of Stephen's little epigrams. I think that's the end. So, of course, it's my lab that did all this, not me. And this is something like the current version of the lab. That the DNA sequence analysis has been in collaboration with Ramin Barakim. I have been funded by ACS, by the Department of Energy at one point, by NSF, but especially by the NIH for 50 years. And so these are all the people in my lab with whom I have published. And that includes 40 PhDs and lots of undergraduates, which are the ones in asterisks. And then there's one other statistic which is really amazing. I counted up all the other labs with whom we have published. And that's this. Okay? So we have been an incredibly collaborative lab. We have found partners to do things that we didn't know how to do, or where we thought we had different perspectives. And it's just astonishing when I go back and look and realize how much of this work has been the rewarding interaction between my lab and some other set of labs. All the way back, well, the first one was David Botstein at MIT in, I think, 1977, and we go all the way to this year with Catherine. So it's been just astonishing a journey for me. And I, of course, wouldn't have done any of it without these really exceptional grad students and postdocs and undergrads, technicians. I should go back and just say that Neil, Sugawara, and Miyuki have been with me for a long time and are really just invaluable partners in doing all of this work. And there have been lots of parties, pumpkin carving parties, and thanks again for the book. Anybody want a question or not? If not, I'm going to show you one other thing. I'll just say two more things. Well, it's not. Michael's not anxious to leave yet. So. When we got rid of the donors, that means we could study something else, which is the end joining process. And actually, the most cited paper from my lab is about end joining. Okay, and we define a lot of the components that are needed to put the ends back together by something called non-homologous end joining. And that was one thing we could do. The second thing we could do, and I'll just say, we got onto a big paper in Nature, which was analyzing tumors in humans, because we saw we had insights about the mechanisms of these end joining events that the guys doing the sequencing simply didn't have. And basically, what we found is, or a conjecture is, that there's a classic end joining process But then there are two other processes which have different dependencies, and we knew those dependencies. One of them is called microhomology-mediated end joining, and the other is called single-strand annealing, and they have different genetic requirements. And we think that that's illustrative of what's going on here. And it is ironic that the protein called RAD52, which is necessary for this process and not this one, has 11 subunits. And I don't know that that's anything other than a cosmic joke, but that's where the transition is between these two things. And I'll leave you with one other. Oh, I'll just say Felix has also done a whole bunch of work on the DNA damage response, and you can go see him. This is a wonderful experiment, and my favorite cytology experiment. The question is, if you make a break, the cells stop dividing. They sit for an enormous long time saying, don't do mitosis because we have a damage. Okay, that's what Felix is going to talk about. So we made a zygote in collaboration with Tim Stern's lab. A zygote has two nuclei in a common cytoplasm. And normally those the two nuclei go through mitosis at exactly the same time. They make two spindles, and they both divide. And one of the nuclei is marked by a green fluorescent spot. And the green fluorescent spot is in a cell where we can make DNA damage with the HO nuclease. So if we make damage with the HO endonuclease, this one divides, but the damaged nucleus doesn't divide, which means that the signal is nuclear limited. And that's something else we don't understand. How does it do that? But I just love the picture, and I'll leave you with that. Yeah?
Oh, that's fun. So the processive enzymes in DNA repair, the exonuclease, the helicase, the polymerases, are those anchored and immobile and like whipping the chromosome around, or is the chromosome mostly immobile and those move along it?
Well, they are not at the nuclear periphery. So in that sense, I don't think that, I mean, the question is, if they're anchored, it's anchored to what? Actin. The answer is we don't know. I mean, but if you allow cells a long time without repairing them, there is that these breaks do migrate to the nuclear periphery to some nuclear pore complex related sites, but their normal repair is not taking place at the periphery, and the exonucleases don't seem to be localized to the periphery. Yeah?
I have two questions related to the -1 frame shift. So do your accessory factors, PIT1 or Pol32, affect the balance between exonuclease and DNA polymerase activities? Or is that a plausible model for what they might be doing in changing?
I can tell you that if we knock out Pol32, we don't change the ratio of +1 to -1. Other than we haven't done TIF1 at all. So.
The other question, I don't think I had time to think about it fully during your talk, but of course RNA polymerase ends up backtracking a lot more than DNA polymerase usually does because of the energetics of sort of strand annealing by making a single-stranded template. So in thinking about the repair fork and the different structure of it, is there a similar energetic imbalance between the product and the upstream DNA?
I think there ought to be, but I don't think anybody has ever really shown it in the way that the transcription people have done so.
Well, then what would make the product be single-stranded, I guess, is the...
Well, it's only...
I'll have to think about it. I'll have to draw it out,
Okay.
I think, to go.
But the copying process is analogous to RNA polymerase extruding its newly synthesized strand. This is extruding a newly synthesized DNA strand and leaving the template as an RNA polymerase unaltered. And so that's. But then this idea that it's falling off means that it's being helped off in some way by something that's dissociating the whole strand so that it can do these crazy quasi-palindrome and these other events. That's what's so surprising, is how unstable is this apparatus.
And so is what you're saying that it's not known what the factor is that enforces the single, that peels off the single-stranded product?
We know that, I would say that from the helicases that I showed, that one of those two helicases, the MPH1 helicase, plays a role in this dissociation process. Anyway, thank you so much.